Thank you. Thank you very much. And it's actually a real pleasure to be here. This is not at all the kind of meeting I usually come to, and definitely I've never spoken at a more of an industry meeting before. So hopefully after this session, I'll have a chance to meet some of you. Actually, somewhat of a painful introvert, so I avoided the speed networking session, but hopefully I'll get a chance to talk to some of you. What I'm going to try and do is, obviously I live my life a bit in this area. I'm the director of Newborn Screening Ontario, where we coordinate and perform the laboratory testing for newborn screening for all babies born in the province of Ontario. That's about 145,000 babies a year, and that's based at our academic teaching hospital in Ottawa. So I'm going to try and give you a bit of a speed tour through newborn screening, how it currently works, how it's currently done, but also where in our field we think it's going, and what is being developed currently in this area. One of the things I'd be really curious to hear about is what we're missing or what we haven't yet thought about, or what are the disruptive issues that are coming from the treatment side of things. I'm a pediatrician, I'm a treating physician, but I'm not in the drug development business. I'm not on the ground floor when it comes to new orphan therapies. So looking forward to that conversation. So what is screening? Someone from our Ministry of Health gave me and a few other quote-unquote learned colleagues a call a while ago and asked us to define newborn screening, and we got into a long debate, and it lasted about 6 months before we came up with something. And this is what we came up with. It's a technical definition, but I thought I'd throw it up there at the beginning. It's a systematic population-based application of a test or inquiry to individuals who do not have symptoms of a specific disease or condition. In other words, they don't have any indication to take them above a population risk at the time of applying the test, in order to identify those who warrant further investigations and or interventions, and that's the key at the end there, is to achieve better outcomes. If you can identify them but you can't achieve a better outcome, then the question is really, what's the point? So where does newborn screening come from? And it really is a story of a rare disease. It's a story of phenylketonuria. PKU was first described by Folling, a Norwegian physician who had two relatives with PKU, 1934. It was the first inborn error of metabolism really recognized as a disease. In 1953, Bickel in Germany described diet, so a severely restricted phenylalanine diet, really a synthetic diet. It's really blurry to me whether this is diet or drug, but a treatment that could prevent the mental retardation, but only if it was applied before the onset of symptoms. So only if disease was recognized in the pre-symptomatic phase. And the third major development was Bob Guthrie in Buffalo, New York, who developed a reliable, simple, and sensitive test. He's shown there with his bacterial inhibition assay, and the little white circles around the central dark area, which is a blood spot, correlated to the phenylalanine concentration in the blood. A really simple test. And his actually, everyone thinks of the bacterial inhibition assay as his major contribution, but it was actually probably the dry blood spot. He invented essentially that technology to take a very small amount of blood from a baby, stabilize it on filter paper, which also allowed for extremely easy transportation to centralized laboratories. We're still using it today. This was in 1961 and is still the mainstay of newborn screening. It rapidly spread through the early 1960s as a public health emergency. I think some of the original estimates that there might be a very large number of institutionalized patients with mental retardation were probably overestimated, but is still an extremely significant cause of preventable severe cognitive impairments. And tied to that was a huge difference with treatment. So this is a truly transformative therapy. And we really don't see the like of this kind of transformation very often. But this combination of events really led to a very rapid institution of population-based screening. If you think about the magnitude of this, we went from babies, being born, having their routine primary care, to a blood sample being taken on every single baby, a whole transportation system and centralized laboratory system being set up to identify these kids with a disease about 1 in 15,000 in our North American populations. So a very interesting story and very well described in this report. It was a genetic testing report done several years ago, but tells the story extremely well. Now, this is my daughter, and I call her my quality assurance exercise. I was following very closely what was happening to her. This was my first daughter. And so this is her having her blood sample taken. It's just to run through quickly what the process is. Dropped onto this filter paper, all her identifying information, some basic clinical information. And I think the relevance of some of that basic clinical information is increasing as we broaden the scope of disease. A little Band-Aid, and this is her having her hearing screen a few hours later. And of course, the idea is to identify diseases early so that you can achieve the best outcomes possible. Now, she did not have any of the conditions, but this is her on her first birthday. And most of the diseases that we're testing for are genetic diseases. And we always have to think both older SIBs, do they actually have an unrecognized form of the disease? And you see that often in the institution of new screening programs. So when we first started screening for an expanded set of diseases, when we first started screening for sickle cell disease, for example, we were picking up older SIBs who had not yet been diagnosed. So you have to think of both older SIBs, and this is my son who's now 9 months old, and you have to think about younger SIBs as well. Screening is about secondary prevention. It's about measures to detect disease before it is symptomatic, and the prevent spread to others is really a public health concept around infectious disease, but also applies, as I just said, to genetic disease. To think about that information and to have an educated decision-making around reproduction. But the key is measures to detect disease before it's symptomatic in order to prevent the consequences, the morbidity and the mortality that are associated with it. A lot of times in therapeutic, when I think about my practice in inborn errors and metabolism, a lot of times we're in this area, tertiary prevention. We have a patient, we have individuals, we have people coming to us with disability, and we're trying to restore function. We're trying to treat it at that time. If I think of patients with Fabry disease who come and they have incipient, renal dysfunction, or they have, a long history of pain missed for many years. We're in really a tertiary prevention mode. We're trying to prevent the next progression of the disease. For many diseases, that doesn't work. I just told you about PKU. Doesn't work for PKU. If you already have a cognitive impairment, that is not reversible. With MCAD deficiency, a defect in breaking down fatty acids, fasting death from hypoglycemia is the presentation. Obviously, you have to have recognition of the disease before the onset of symptoms. So you have to be operating in a secondary prevention model for those kinds of diseases. And the story of screening is a story of figuring out which diseases fit really well with the secondary prevention models. If you have not read this and have any interest in screening, it's available on the WHO website. It was written in 1968, and in my opinion, is still the seminal work in this area. It goes through and describes the principles and practices of screening for disease, and is often thought of as the criteria for deciding whether or not a particular disease or condition should be targeted by newborn screening, or any type of screening. And I just highlighted the two key words here, treatment and early. And again, it's that concept of secondary prevention. Chris mentioned, and this morning I think we heard about natural history. Do we know the natural history of disease? And it is especially important when screening. If you have someone who's coming to you with symptoms of a disease and you're diagnosing a condition in that context, you already have an idea of where they're at in their natural history and what the likelihood is that they're going to progress in a certain path of expected issues arising with that diagnosis. I've had the experience a few times in kids who I know have a disease but who have never been sick, and you're communicating with the family and they're saying, how do you know my child, I don't think my child has this disease. How do you know? And it's really a key question. If we're not able to predict the natural history of the disease extremely well, we risk starting therapies that aren't needed. We risk raising alarms for families that don't need the alarms raised. we also risk losing the faith of the parents who do have a child who then does go on to become sick. So this concept of knowing and being able to predict the natural history is very important. So I'm not going to go into the details here, but this is from 2006. Many of you are probably familiar with this, but this was an exercise by the March of Dimes and the American College of Medical Geneticists, recognizing that PKU, it's great, we've had a few other targets of newborn screening, but what else should we be targeting for secondary prevention by newborn screening? And through their method, and there's a great debate in the literature that has ensued in the couple of years after this as to whether their method was great or not, but it was the first one that was done and done rigorously, and I think has been extremely influential. They came up with a core panel of 29 such conditions where the impact of early treatment is variable, but it is not debatable that there is an impact of early treatment. The Secretary's Advisory Committee on Heritable Diseases, I think I got that right, it's a long name in the United States, I think is still extremely influential internationally. I know they're very influential in Canada through their process to consider what diseases should be added next to a newborn screening panel. And this is from their website. It's updated periodically for the conditions that they've considered, the ones that they've performed a substantial evidence review, and the ones that they've actually recommended to be added to their RUSP, the Recommended Uniform Screening Panel. And a few 22q11 deletion syndrome, they decided not to go forward to an evidence review. They felt there wasn't enough prima facie evidence at that time, but came back with recommendations as to what was lacking in the evidence, and that has provided scientific guidance. So there is a robust process in place in the United States and in many other countries. And I love this slide. Oh, I move things around, but the circle here was supposed to point at this person behind here who's me. And this was the premier of our province when we launched. And just to say that despite, process, which is, I think, exceedingly important and very useful to maintain societal buy-in, to make sure we're doing the right thing, the decision of whether or not to screen often is political as well. And I just wanted to highlight that here. So I just want to move quickly on to some comments on technology. I mentioned the BIA, the bacterial inhibition assay that Bob Guthrie developed. One test for one disease, a single marker, semi-quantitative, manual, slow, and a high, relatively poor performance. In the 1990s, Dave Millington, Don Chase, many Americans were involved in the institution of tandem mass spec technology for really multiplexing the screening tests for many inborn errors of metabolism. And this was an example of technology driving progress. We moved from this type of paradigm to a paradigm of testing where one test could detect many diseases. You could look at many markers and ratios. You were looking at quantitative versus qualitative. It was automated, fast, it was more robust, and better performance. So this is really the current state. We still have this concept of one disease, one test, one biomarker, one treatment, but we moved in the 1990s to multiplex, still biochemical tests, multiple biomarkers, multiple diseases, and multimodal treatments. And I think in the screening world, often the impact or the relevance of the treatment developments in the treatment world may be underestimated. So congenital hypothyroidism, where the body is not able to make thyroid hormone, maybe because the thyroid gland isn't there, or there's an inborn defect in thyroid hormone biogenesis, is still targeted in that one assay, one marker, one treatment. I just mentioned tandem mass spectrometry using the example of amino acids, but acylcarnitines as well, but 40 different biomarkers that can target many different diseases. This is also real time. This is really a testing strategy that has developed in the last 5 to 10 years into routine practice. The concept of tiering. If you have relatively poor performance on a first tier screening test, it may not be adequate on its own to implement for a screening program. But what if you had a really specific test, but it was too expensive, or it was really complex or very time consuming, and you just can't run through, our lab, we're running through 150,000, 160,000 samples a year. You just can't do that. Well, what if you could filter out at high sensitivity using a first-tier screen within the same lab, within the same sample, no contact back to the family to raise false alarms, et cetera, and tier the tests so that you can achieve an overall screening performance that is much higher. And the earliest example, one of the earliest examples of that was really the IRT CF-TR mutation panel system, first piloted in Wisconsin for CF screening. Still very much in use today, and this is an example of a first-tier, second-tier system. The positive predictive value, so the probability if you have a positive screen with only one mutation is still really low. It's 2%, and that is a problem with this kind of a testing strategy. And I'll mention later one of the ways to deal with that might be a third-tier strategy. So current issues. Consent is a current issue. When you have multiplex panels, parents may want to consent to some, but not all. Incidental findings. How do you deal with things that you find that you weren't necessarily looking for? Compromises for different targets. So the more you add, you have a finite sample that you're taking. The timing of the best to best screen for those diseases may be different for the different diseases. How do you make those compromises? Premature infants are different, and I'm not going to go beyond that, but they are different. And they're a small, say 7% of the newborns, but they account for a significant percentage of the problems. And disease-specific tolerability of false negative screens. I think if we were ever to miss a child with PKU in our screening program, I think that would be absolutely and completely intolerable. But what I showed you with the CF screening strategy, if we don't have, our mutation panel is not going to pick up every single child with CF. But that delta of treatment, the expectation with CF is not that we will pick up every single child. We pick up probably about 98% of them. But there is a different toleration of false negatives. So what's happening in the future? I think I've mentioned dry blood spots. I think there's different types of samples that can now be collected, and that may actually be less invasive. I mentioned fetal DNA and maternal serum. If that technology, or if fetal cells in maternal circulation allows for single gene disorder testing, some of the aggressive newborn onset diseases that we have trouble picking up fast enough by newborn screening methodology may be amenable to earlier diagnosis in the late third trimester, for example. So that's an earlier concept. Later, what about diseases that don't present in the newborn or early infantile period? Adrenoleukodystrophy is an example of a disease that presents usually in later childhood. Is the right time to screen in the newborn period, or should we be screening later? And then next-gen sequencing we'll hear about next. Point of care, I think, is a huge one. So the Star Trek tricorder, I think we're heading there. How do we deal with that? And then further advances in metabolic testing. I'm not going to dwell on this, just to say that there are many dimensions to technological advance that we should be keeping in mind. So CF, I already mentioned. Third tier, CFTR sequencing is something that I think is coming very quickly, already in place in some places, as a way to deal with this issue of very poor positive predictive value with the one mutation testing. And then this is one that I think everyone is talking about. So the paradigm has been for a long time multiplex biochemical testing followed by definitive molecular genomic testing. And I think we may be seeing a time where that's going to flip on its head with first-tier genomic screening, which may allow you to to target a much larger panel of very rare disorders. And followed up, I think you are still going to have more sophisticated biochemical and functional testing in order to better delineate the pathogenicity of the mutations you're finding, to better stratify risk or expected natural history. And just to mention that there's a couple of diseases that we won't be able to target using next-gen sequencing. It's not a panacea for all things. A couple other paradigms to mention. Pharmacogenomics screening for risk, and two examples, pharmacogenomic screening and type 1 diabetes screening, where the concept is really having the right information at the right time. This is a paradigm that's in investigation, is being piloted. Screening for acquired disease is another concept that has not been in the newborn screening world. Infectious disease may be in this area, celiac disease. And then screening for intervention research, especially when you have diseases where secondary prevention we know is going to be the only way to change outcome. So spinal muscular atrophy to me is always a really good example. If those motor neurons die, you can't bring them back to life. The therapies that are going to work are going to be ones that prevent those motor neurons from ever dying. And this is going to raise even further issues. And this is really the stage we're at now, is how do we apply these technologies and deal with these issues before instituting them into practice? Education and consent. Again, I'm not going to dwell on that, but I don't think the era of all or none panels will be able to persist. Prediction of natural history, I've already talked a bit about that. Surrogate outcomes. What are we going to be able to use to trial new therapies? And measuring our long-term performance. How do we know how well we're doing? Are we really making a difference in the health of the individuals affected with these diseases? CIMDERN is a Canadian Inherited Metabolic Disease Research Network, and that's me and my colleague Beth Potter set that up because we realized the only way to do this is in an observational research method, to really know, are we making a difference to those kids with inborn errors in metabolism? So quickly back to PKU, we do have an excellent biomarker. It's a great predictor. Phenylalanine is a great predictor of the natural history. If you have very high phenylalanine levels, you are at very high risk of cognitive impairment. If you have low levels, you are at very low risk. It's also an excellent surrogate outcome measure to use in evaluating new therapies, such as PEG-PAL, currently being evaluated, and Kuvan, which is in market. To point out, this is the exception rather than the rule. Such biomarkers are not a panacea and often are not available. And patient-centered and other clinical outcomes, I think, are going to be very important in this endeavor. So a second quick example. This is a pilot that we have ongoing right now with our infant hearing program in Ontario. About 60% of kids who are going to have prelingual, so before the onset of speech, hearing loss, are deaf at birth. About another third to 40% will develop that hearing loss in the first year of life. But we don't pick them up when we do the hearing test in the first week of life. So is there something we could do to try and pick them up? So our pilot is really looking at CMV infection, cytomegalovirus, which is the leading cause of prelingual hearing impairment, as well as mutations in a select number of genes to identify a cohort for high-risk hearing surveillance. And I seem to have lost my last bullet on the end there, which was the intervention that is planned is for hearing augmentation, communication therapies, learning sign language. Those are the interventions that are planned and are very effective in maximizing developmental outcomes, cochlear implants as well. But what if there was an orphan drug that could treat, prevent, mitigate the hearing impairment, but only if used before the onset of symptomatic hearing loss? And this really is the paradigm that I think we have to have our eye on. If I broaden that statement, what if there was a transformative treatment that could treat, prevent, or mitigate the morbidity or mortality associated with a rare disease only if used before symptomatic disease? That's really what we're talking about when we're talking about screening. Better outcomes from starting treatment early in the course of the disease and finding children with rare disease during their window of opportunity. And as usual, all of these things exist in a network. This is just our network in Ontario. And if I think of newborn screening programs internationally, we have several that we work with in other places. These are the treatment centers and hospitals where kids obtain their care. Academic institutions where a lot of this research is being run through. We're at the University of Ottawa. The CDC has a very active role. Registries, policy shops, government, research funding agency, and industry. These are all the people in our lab and our program. We have a big social media strategy that a couple of our genetic counselors pushed me into, and I think it's a really good idea. I'm the introvert, so they have to push me into this kind of stuff. And that's it, I guess. Oops. Oh, I had one last slide, which just had, I think I had one last slide. It's all right.
Pranesh, thanks for the presentation. If you could, thank you.
The last slide had more pictures of my kids.
Yeah. So we're running a little bit over time, but just one question for you. So our company has a drug for Duchenne muscular dystrophy. It could be the first disease-modifying therapy approved, but it only treats a small percentage of boys, about 13%. Can you articulate as a clinician, pediatrician, how do you deal with those issues where you might have a newborn screen for a disease and only a very small percent might benefit from a treatment option? Or would you be able to articulate why newborn screening might be helpful even when there's no real interventions available?
Yeah, so two different questions there, and I think the first one is fascinating. It's really an example of personalized medicine. Is disease nomenclature, what is the disease? I mean, this gets into philosophical stuff. Is Duchenne muscular dystrophy the disease, or is it a mutation in the DMD gene, which is the disease? And we have the same thing with CFTR. I actually glossed over that. I had a bullet point in there about mutation-specific therapies. Certainly what screening is about is identifying individuals in a population who will benefit from early treatment. So if you know that boys with Duchenne with a massive deletion are not going to benefit from your drug and there is no other treatment, then I think you got to really question why you're screening for that condition. It may be for reproductive benefit. It may be for early identification and onset of supportive treatments. And we have a whole strategy in Ontario that's just starting on non-disease targeted screening for developmental delays. And my job in that is to add the biological element. So there is a benefit to picking up a kid with incipient developmental delays at 2 months of age rather than at 6 months of age, and to institute those interventions. But if you could find that kid at 2 months of age and recognize that they have a treatable inborn error of metabolism, or they have a mutation that in whatever gene that has a mutation-specific therapy, well, then you're really getting to the personalized medicine. And I still like that term. I know it's going out of vogue, but it's the personalized medicine promise that you're really going after.
Thank you very much.